Okay, we can identify two regions in a gene. And these two regions, they have names. One type of region is called exon. One type of region is called introns. Again, introns are non-coding. That means within a gene, there are non-coding region. Within a gene, there are non-coding region. So exons are coding region, introns are non-coding region. Then why these things are existing like this? And at least, okay, what is the function of intergenic region? At least we can highlight one functions. They have regulation. They regulate the gene expression, can be regulated. Then why exons and introns they are in the genes? What is the functions of introns then? And how does this operate? Right. We try to look a little bit what happened during the protein synthesis process. However, protein synthesis process, we are going to learn in this section in detail. But before we're going to learn protein synthesis, I will give you a summary of protein synthesis to explain the role of exon and introns. So exons are coding region and introns are non-coding region. Now I will give a summary of protein synthesis from gene to protein before we come into the detailed lessons. Okay, for that, I again show you this region, this. So this is a gene and it has two regions. Okay, here it represents 3 exons and 3 introns. So here is exon, here is exon, here is exon, and here intron, intron, intron. So this is exon, this is intron, exon, intron, exon, intron. So I just draw in alternatively, but you know they are randomly organized. It's not a tree like this. There are many different exons and intron. The organization is very random, so we can't say it is always like this. So they are randomly distributed exons and introns. Now this is the DNA molecule. During protein synthesis, DNA should be copied as DNA, just the nucleus. Okay, that's a nuclear membrane. Because DNA is inside the nucleus. Now the first step is the whole sequence, exon and intron, copied into the RNA molecule. So this is copied. And this is called pre-mRNA. The first step is copying. This copied process is called transcription. The process is called transcription. DNA copy into the messenger RNA. Now, messenger RNA has exons, introns, exons, introns, exons, introns. So it has again complete length of the gene in the pre-mRNA. So this is clear.
So it's copied by the mRNA molecules, no, sir?
Copied to, the copied molecule is now called mRNA, messenger RNA. DNA, the sequence is copied to the mRNA molecule. That means base sequence copied to the mRNA molecule.
Yes, clear.
Okay, in the next stage, there is a process take place because introns are non-coding region. They are non-coding region. They can't make proteins. The introns are not designed to make the proteins. They can't make protein. If they make the proteins, the protein is completely altered, different proteins. That doesn't work. Then in the next step, post-transcriptional modification take place, right? The second one is called post-transcriptional modification. In post-transcriptional modification, introns removed from this pre-mRNA molecule. So introns removed, and then exons. Now this is one exon, and this is one exon, and this is one exon. Let's say A, B, or C. A group of enzymes called spliceosome cut interface of intron and exon. At this stage it cut, and here it cut, and here it cut, and like this, they start to cut these intron and exons. And after cutting, it can only join exons. Spliceosome group of enzyme then join these exons. So when it join, we get a short now sequence like this. This is a mature mRNA. This is called now mature mRNA. Now, one important significance of this, if we remove introns, there is a chance of making different combinations. For example, if this ABC, it can make ABC, so it can make another sequence like this, B, A, C. So BAC is different sequence. Or it can make only B and C. Now, in the post-transcriptional modification, there is a chance it can again make different sequence. It can make many different sequence by alternative combinations of the exons. Now, why is that important? Human has 25,000 genes. Human genes are 20 to 25,000 range of genes we have. But if you look how many proteins we have many different proteins. The proteins are not 25,000. If you take a slight change of the proteins, probably we have millions of billions of proteins. So when you learn immunity, one antibody is a protein and that antibody is different to another antibody. Then we can say there are two different proteins because antibody binding sites are different. They have a different amino acid sequence, shaped different. As a group, they are one protein antibodies. But within antibodies, we have billions of millions of different antibodies. So how this is possible? This is possible to make many proteins. So I just write billions of proteins, different proteins. The billion is not correct word, but just to say more than 25,000, the proteins are many. If I use many, it could be less than 25,000. So just give the word billion, understanding many billions of proteins are there. This is possible by this kind of different combinations. That's why introns and exon existing in eukaryotic genes important to produce this concept. Actually a important concept which is deleted from the old syllabus to new syllabus, because the molecular biology like things are advancing day by day, but they just put very basic things, removing the important thing. That concept goes as a one gene many proteins, one gene many proteins. So one gene many protein is a very important concept in molecular biology, and this is not existing here. Just say introns and exons are there. After that, what is the important not mentioned? Now you understand introns are exons are very important. Because of that, human has 20 to 25,000 genes, but we can make many billions of slightly different protein structure because of this combination of different exons during the process of post-transcriptional modification. Post-transcriptional modification. After that, the mature RNA leaving from nucleus to the ribosome. And from the ribosome, mRNA provide the codes to make the polypeptide chain. So then inside the ribosome, the polypeptide chain is developing. That's the gene expression and protein synthesis. The whole process of gene expression is copying into the mRNA and make proteins at the ribosome is called gene expression and protein synthesis. Now, what I wanted to highlight when you talk about the eukaryotic gene, we have a non-coding regions called intergenic region. Intergenic regions are, one function is they have some kind of a regulation of the gene expression. When it comes to the genes, again, two regions, exons and introns. Exons are coding region, introns are non-coding region. Why introns and exon exist, we can explain by this process called post-transcriptional modification. They cut intron and exon, introns removed, exons joined together in a many different way, can make different proteins. So is this clear?
So in the transcription, so is the uracil coming so there?
Yeah, that's the basic rules are there. That the RNA molecule, they don't have a thymine, they have uracil. So we're going to learn in detail that one, because in the next protein synthesis process, we're going to learn every single step detail, but we are not learning about this kind of process like introns and exons like things. But I just highlighted here intron and exons. We are learning nature of eukaryotic and prokaryotic genes. So we try to understand how they are differ and what are the significance of prokaryotic and eukaryotic genes. But when it comes to protein synthesis, definitely we're going to learn a detailed process. Okay, next thing is the prokaryotes has very special method of copying this gene. And here I mentioned as one change that, okay, another thing. So prokaryotes, they have a genes. And as I told you, non-coding regions like intergenic regions like things are not much prominent in prokaryotes. They have always continuously genes. Now, when it comes to genes, we have again question, do they have introns and exons? So what is your answer? Prokaryote genes, do they have this exon and intron concept?
Yes.
Well, it's like if we divide again precisely domain eukarya, archaea, and bacteria. Bacteria, archaea, eukarya. Yes, eukarya, definitely yes. What about bacteria?
They have.
They have archaea.
Don't have.
Okay. So I checked something that is how far familiar with the resource book. Okay. I just take this one. Now in unit 2, I take these tables. The comparison of archaea, bacteria, and eukarya. Now it says introns in gene bacteria, very rare. And present in some genes in archaea, and okay, eukarya, yes. This is archaea. I think we try to remember as it is directly from the resource book, very rare. Present in some genes, present in many genes. But the correct data is no evidence of these introns have found in prokaryote despite the sequence of more than 100 genomes. And scientists came up, we have done so much of study, but we never find introns in prokaryotes. Current data propose there is no introns in prokaryotes. However, the resource book doesn't say that. It says very rare, present, and many. For the sake of getting marks, so we should adhere to this very rare, present, and present in many genes concept. Because you check the table in unit 2, all prokaryotics, archaea, and eukarya molecular level differences described. When it comes to molecular level, DNA is important molecule. They have a comparison of this, and here we should also highlight it again in case very rare in prokaryotes. Okay, next, we are coming to this concept of the prokaryotic gene expression. Now, prokaryotes, gene expressions happen like this. This is one gene, and this is another gene, and this is another gene. So we can see three genes at the same place. Three genes behave at same way, and they operate in the same way. As a result, this kind of a collection of gene in prokaryotes called operon. So what is operon? So this kind of a collection of gene or cluster of genes, collection of gene at one place in bacteria is called operon. I think the operon word is important because we use in biology this concept to explain many things. As a result, this become one of the most important thing in biology, the operon word. And when it comes to the gene expression, what's going to happen? This make a mRNA, this make a mRNA, this mRNA. So this is the mRNA. And this make a protein. This is going to make another protein. This is going to another protein. Protein 1, protein 2, protein 3. So this is the well-known method of gene expression, organization of genes in prokaryotes. So we do not going to learn detail about this. Otherwise, we can understand much good biological concept with this operon, but only the word is used operon and the definition is given in the resource book. No point of learning details about this prokaryotic gene expression within this operon. But just when it comes to the eukaryotes, this is the gene, and I should always add something onto that. There's a region before that, and here there's a region before that, mostly serve as a regulatory site. So this is the regulation region. So gene has a regulatory region, and the regulatory region, we can also find different components. So these different components need to regulate this gene expression. We can learn it later when we are doing protein synthesis. We can learn what is the exact role of regulatory region. In this case, here is mRNA and the protein. And this is prokaryotes. So here again, we learned the expression of genes or organization of genes in eukaryotes and prokaryotes. Okay, so this is done. The nature of prokaryotic and eukaryotic gene expression is also completed. So if you have any doubt, you can ask.
No, sir, no doubts.
It's raining, finally. Here. Next, we are now going to move from Mendel hereditary factors to chromosome theory of inheritance. Because Mendel used hereditary factors to explain his theory of genetics. Now, this Mendel concept did not reject at any time, but Mendel concept improved in many times in the history. To explain Mendel genetics, scientists further studies his law of genetics, and with the discovery of genes, chromosomes, DNA, his theory is further getting stronger and stronger. Now, this chromosome theory of inheritance still not a new thing for us, because we have learned this in genetics without highlighting chromosome theory of inheritance. We understand it because we did not use hereditary factors, because if we use the Mendel words and everything is very difficult to learn. We directly use chromosome, genes, alleles like things. Actually, that is a chromosome theory of inheritance. But before we come to that, 1902, a scientist Boveri and his groups and his colleagues, they actually proposed, look, Mendel explained like this, but what he explained scientifically, it is like this. There is a gene on chromosomes, and these chromosomes separate, and the gene can also separate into gametes. And after that, gametes get separate genes. When they fuse together, again the homologous chromosome pair up, and the gene come up to express. So he used the terms chromosome, gene, allele-like things to explain the Mendel theory. So this further modified to become chromosome theory of inheritance. And together with another scientist, his name is Walter Sutton. So they established chromosome theory of inheritance. So chromosome theory of inheritance, it's same thing what we learn in genetics. The diploid cells replicate, and after diploid cells replicate, chromosome number doubled, and during meiosis 1, reduction division happen, and then a homologous chromosome separate, and at the end meiosis 2, gametes form. So then it get the half a number of chromosomes. So that's actually the chromosome theory of inheritance, which is not new thing. Only thing is we did not use this term chromosome theory. Otherwise, you can explain, okay, this is a cell with a homologous pair of chromosome. What happened during gamete formations? Okay, this chromosome number double. That's chromosomes with the chromatids, sister chromatids. And then after that, meiosis 1, they are separate. Meiosis 1. And then meiosis 2, it form again the gametes with half a number of chromosomes. That's a replication, right? So this is a diploid cell and then make a haploid cell again. So we have learned this in meiosis. We have learned this in genetics. So this concept actually came originally from the work of Boveri and Sutton, and they proposed this as chromosome theory. And we are very familiar with this chromosome theory, even though we haven't used this term, chromosome theory. So it is all collection of meiosis. If you go and refer to meiosis, all the stages, plus genetic, we have learned how it really happening when passing information from parents to offspring. Now, why this theory get more and more stronger after that? So when the 1902, around when they proposed this theory, it is new to the scientific world. For us, it's nothing because we learn meiosis from the very beginning. The whole process we learn. But when they proposed 1902 around, this is really interesting things to understand so many things. So Mendel used the plant, pea plant, Pisum sativum plant, and he tried to use a certain existing phenotype, how pass into the next generation. Now, when they proposed this chromosome theory of inheritance to scientific world, this got a lot of attraction, and many scientists, they try to use this theory to explain their concept. So we have so much of evidence. I mean, we don't want to go to that evidence to see one by one. This Bavarian Sutton chromosome theory of inheritance, proved by other scientists that time, yes, that's very correct what they say, because I observed something like this, and it is exactly happening according to this. So one of the example came up to explain this concept. A physician called Archibald Garrod gained close to same year, so that is excited with this kind of theories. Now he tried to use this theory to explain how genetic diseases, some kind of a disorders passing from parents to offspring. So he used this theory then. So that is completely new than that time. So offspring born with a genetic disorder called alkaptonuria, they have an inability to metabolize certain compound called alkaptone in urine to metabolize product. As a result, alkaptone can oxidize to make a dark color, like a black color. During get black color when it exposed due to inability to enzymes to break down this alkaptone. So he studied this disease and he found like pedigree diagrams. So he checked these pedigree diagrams affected like here, and they look the parents probably one is affected like this. So something like this, he just go with the pedigree diagram to check how this is possible. And then he found this is coming from genetically, this family background, this is coming genetically. And then he used this theory of chromosome inheritance. He can explain well the disease. Okay, it is like this, like this, like this. So it become a very interesting theory for scientists. And also, this is one example. The concept of chromosome theory of inheritance getting stronger, and it is proven by many other scientists taking example like this. So if we think about beginning of 1900, so if we there in 1900, these information are really big information for them. But now we passed all these things. For us, it's nothing now because we know everything. But that time they have no idea about these genes, chromosomes, and how it pass into the next generation to next generation. Even you can understand DNA molecule, structure of the DNA molecule discovered in 1952, 1950, this 1952 around. And then only they try to explain how the genes looks like and how it pass into the next generation. But that time for them, they at least came up with the chromosomes and genes, how they behave, and how the information is passed into the next generation. So this is a starting point. So they use this theory. But do you know that in this period, 1900, many scientists refuse to believe DNA is the molecule which is responsible for heredity. They say protein is the molecule which is responsible for heredity. No one proved at this time, and scientific community refused to accept the heredity is it's the DNA molecule until 1950 around, when they discover everything. Then the scientific community start to believe heredity, DNA molecules, and informations, all these things. So the period from 1900 to 1950 is one of the best period for scientists to identify the DNA or protein, which one is the real molecules. That's why learning biology in history, certain things are just simple thing for us. We know these things. But for that time, scientific community is a big thing, and they would like to use this kind of a theory for the further development. So one by one, they used to develop science that time. So all single information important for them until discovery of the structure of the DNA molecules. So that's it about the chromosome theory of inheritance. 1902 around, Boveri and Sutton explained this, and we know it is something what we learn about meiosis, whole concept is going to explain about the chromosome theory. That time, people, it's good for the people, scientists, to use the theory to explain many things. For example, this alkaptonuria genetic disorder can be explained by chromosome theory of inheritance. Okay, with that, we stop. We have a brand new lesson coming up, protein synthesis. Genetic code and protein synthesis. Next time, we're going to learn protein synthesis and genetic code, right?
Yes. Okay.
Okay, then see you next time, right?
Okay, sir. Thank you, sir.
Okay,
Bye.
bye.